the organizers, and thank you for being here. Good afternoon, everyone. My name is Tingting. I'm from Stanford University, and today I'm going to talk about our design for chemical reporters based on cephalosporin for Mycobacterial tuberculosis detection. So a little bit of background. It has been estimated that by 2050, superbugs can be responsible for 10 million deaths.
Actually, you are getting some sound disturbance. Can you just check, if you are using earphone and AirPods, can you check once? Yeah.
Sure. Is it better or
yeah, it's
worse?
better.
Okay. Should I?
Yes, you can.
Hold on. Okay. Did you hear the previous slide, or should I go from the beginning?
You can go from the beginning. No issue.
Okay. Yeah. So today I'm going to talk about the design strategy that we use to synthesize the chemical reporters to study Mycobacterial tuberculosis. So a little bit of the background. So it is estimated that by 2050, the superbugs is going to cause 10 million deaths worldwide. To give you a better understanding of the number, we can compare the death toll with that of the COVID-19, influenza, and the cancer. And this number even exceeds that of cancer. So specifically, if you look at the tuberculosis, so tuberculosis remains one of the deadliest infectious disease in the world, claiming 1.5 million deaths in the globally, especially in the developing countries. If you see all the very dark red regions, and it's although it's a very ancient disease, it's still a very serious public health threat. So this disease is caused by a bacteria called Mycobacterial tuberculosis. And this bacteria is infect people and spread around the populations by air droplets. And the infection begins with this inhalation of aerosols that contain the bacteria, which are primarily taken up by the alveolar macrophages. Inside the macrophages, Mycobacterial tuberculosis is usually found within the phagosome of variable maturation states and encounter a variety of host defenses, including phagosome acidification, antimicrobial peptides, and reactive oxygen species. This range of distinct microenvironment exerts discrete selectivity pressures on individual bacterial cells, which suggests that the Mycobacteria need to adapt this environment to conditions to survive. Because of this complicated intracellular lifestyle and its importance in studying their growth and divisions, it's very important to kind of design chemical reporters to study their behavior in the innate environment. So current imaging strategies include genetic labeling, and it's very common for people to study intracellular pathogens with genetic reporters that express fluorescent proteins, for example, in as you can see in these pictures. But the problem is that the long half-life of these fluorescent proteins can limit the real-time imaging of their behaviors. And also the genetic modification could reduce the virulence of these bacteria, thus limit our study of the innate behavior of the bacteria. And in addition, genetic labeling cannot report on cell wall biosynthesis, which are very important to design new antibiotic to fight the disease. Another approach is chemical reporters that label the Macrobacterial Glycans. So if this is an image of Mycobacteria cell wall structures, and as you can see, they have very thick cell walls, including Mycobacteria membranes, paraplasm area, and also cytoplasm membranes. Together, this form a very formidable layers to for the bacteria to maintain its shape, and also able to do a lot of biosynthesis to degrade the antibiotics. So people have been how to synthesize these macromembranes is by using these sugar building blocks, and by the help of this enzyme called H85, they can attach a lipid tail into one of the sugar molecules to anchor the building block into the outer layer of the bacteria. And in this way that they can form this very thick layer of macromembrane. And people have been, based on these building blocks, they have been synthesized a natural sugar molecules called, based on this sugar called trihalols. And they attach a dye, fluorescent dye, onto the sugar. And then supposedly that these sugar mimics will mimic the behavior of the natural trihalols monomethylate and being incorporated into the cell wall layers using the same biosynthesis pathway. So this has been a very popular method to label the bacterial cell wall and study the bacteria. However, the problem is that as you can see, that these metabolic labeling could require very high working concentrations. And also this method is not specific to this type of bacteria, because the enzyme that this group are using is conserved in all of the actinobacteria phylum. So, so this is consists of a group of a variety of disease-relevant bacteria. So this probe could cause some toxicity when you study the bacteria in the macrophages because of the high working concentration, and also it's not specific to this just this Mycobacterial tuberculosis. So what we think is that what I have been discussing is called always on probe. So that means that these probes are always giving the signal before and after they encounter the target. So this could lead to some unspecific signals, and also it doesn't give you much of the information on the activity of the bacteria. So there's also another type of probe called single log probe. They can only be activated and giving out the signal when they encounter the stimuli. This gives you some specificity and sensitivity, but we thought that maybe we can add another layer to this type of design strategy by giving it a double locked probe, so that only encounter one stimuli doesn't give is enough to give the signal, but only encounter the second stimuli, and together they will give the signal. Thus, in this way, we hope to achieve higher signal to background ratio, and also give you another layer of selectivity when you detect the bacteria. So in this case, that we in order to have the two log, we need to look for another biomarker. In this case, when we still looking at the cell wall structure of bacteria, we turn over attention to an enzyme called Beta Lactamase. So what does this enzyme do? So to understand this enzyme, we have to understand the bacteria cell wall structure. So in this area called the paraplasm, there's an enzyme called the penicillin binding protein. This protein's job is to cross-link two small peptide in order to form this very thick and strong layer to hold the shape of the bacteria. So people have been mimic this this structure of peptide by designing antibiotic called beta lactam. This type of antibiotic is one of the most popular antibiotic still in the clinical settings. And this type of antibiotic will trick the penicillin binding protein to attack the antibiotic instead of causing the peptide, native peptide to form the bacterial cell wall. But the bacteria is very clever, and it's developed a resistant mechanism called by expressing the enzyme called beta lactamase. So instead of using the antibiotic to trick the native protein, penicillin binding protein, this type of enzyme beta lactamase will attack the antibiotic and thus rendering this antibiotic useless. So over the past few decades, this kind of there's a war between the bacteria and the human beings. So human has been able to develop different antibiotic that cannot be hydrolyzed by the beta lactamases. But bacteria, on the other hand, also develop a range of different beta lactamases, as you can see in this table, to actually hydrolyze the antibiotic. So we thought to use this mechanism by attaching instead of by using the structure of the antibiotic and attach a dye on the 3 position of this this molecule. So upon hydrolyzed by the beta lactamase, the dye will be released, and then we can see the signal. And this strategy has been used in the past by as you can see here, that by attaching a blue emission dye, and then we can understand the activity of this type of beta lactamase activity behavior, and thus achieve the bacterial detection. So this has been reported, and we sought to use this scaffold to further design of a dual targeting probe. So in terms of Mycobacterial tuberculosis, they are naturally resistant to beta lactam type of antibiotic by expressing the beta lactamase C. This is a very unique type of beta lactamase, and it has been reported that by doing some structure modification by attaching a cyclopropene group on the 2th position, and also O-methoxy group on the 7th position on this cephalosporin ring, we could achieve a more specific labeling of the MTB. So in this case, that we can attach a dye on the scaffold to do the reporting signal as a reporter, and also attach the first recognition site, which is the beta lactamase recognition moiety. And then by attaching a second recognition group, we could achieve a further and more specific detection of the bacteria. In this case, that this work has been demonstrated and reported in the 2018 work. And indeed, we achieve more specific detection of the MTB. But when we doing using this probe in the intracellular environment, we see a lot of non-specific background. As you can see here, that the bacteria is labeled in the red, and then the probe is emitting green signal. And when you incubate both the probe and the bacteria in the macrophages, you can see that it has a lot of non-specific background, and we could not detect the specific signal for the bacteria. So this probe is not ideal, and we sought to further improve this design strategy by changing the second targeting group using trihalose, because we saw that by further tuning the hydrophobicity and hydrophilicity of the probe, we could achieve a better targeting ability of the intracellular pathogen. So in this case, we used the similar dye to do the reporting, and then the similar beta lactamase recognition moiety. But then we changed the second targeting moiety into the cell wall structure, the trihalo's sugar. And then after hydrolyzed by the beta lactamase to release the probe, we saw that this probe will be further anchored to the cell wall membrane of the bacteria, and thus achieve a more signal to background ratio because of the abundance of this trihalo sugar on the cell wall. And then we linked, then we put all of these elements together with alkyl linkage. So, and again, that the probe we thought to work in this case is by first it's not fluorescent, and when they incorporate into the macrophages and target to the bacteria, it will be first hydrolyzed by the beta lactamase to release the fluorescent sugar molecule, and then further processed by the enzyme H85 to attach the lipid chain, and then anchored into the outer membrane of the bacteria. And then we can achieve first more specific, and second higher signal to background ratio of the detection. So in this case, we select the trihalo's trihalose sugar for the anchoring molecule because of the synthetic flexibility, that we could use quick chemistry. And this molecule has also been reported in the past study. Further, this it has been reported that by modification of a different position on this sugar molecule, you can achieve different labeling efficiency of the bacteria. So in this case, it has been reported that the 6th position and the 4th position has been showing the highest labeling efficiency. So similarly, we want to test that in our system to see that if which position will give us the highest labeling efficacy before we put everything together. So we first attach the fluorescent dye onto the 4th and 6th position, and then incubate them with a variety of fungi and bacteria species. So in this case, the C. albicans and E. coli are kind of remotely related to Mycobacterial tuberculosis because they're this one is fungi and this one is gram-negative. Asaurus is gram-positive bacteria. So those 3 species are kind of remotely related to the Mycobacterial tuberculosis. Although they are clinical relevant, they are not showing any signal. C. diff and MSMAC are more closer related to Mycobacterial tuberculosis, and because they have the cell wall structure, as we discussed in the previous slide, they all show great fluorescent signal. So this demonstrates that our probe indeed can distinguish the Mycobacterial tuberculosis with the rest of the bacteria species. And also it shows that if the those trihalose probe is not specific, because if you have the cell wall structure, you can show the signal. So further, we quantify the signal intensity by using a variety of, we quantify the signal with flow cytometry, and also we demonstrate that within 2 hours that the labeling efficiency can reach the highest. And also the 6th position give you the highest labeling efficacy. So further, with that understanding, we further attach the cephalosporin structure onto the dye and also onto the sugar, and then compare the this molecule with the one without the beta lactamase recognition moiety. So in this case, similarly, we incubate these both of the probe with a variety of bacteria species. So different from the previous probe, we noticed that because C. diphtheria doesn't have the beta lactamase enzyme, it doesn't show any signal, although it has the same cell wall structure. So in this case, we come up with a conclusion that because of the two targeting moiety, we were able to achieve a higher and a better selectivity towards the Mycobacterial tuberculosis than over other species. So further, we want to understand if this probe is using the same biosynthetic pathway as that of the trihalols. You did a computation study by incubating the probe with a high increasing amount of free trihalols. So with a higher amount of trihalols sugar, you see a decreasing of the signal. And this demonstrates that our probe is using the same biosynthetic pathway as that of the free trihalols sugar. So further, we want to understand if our probe will be able to enable MTB labeling for phagocytosis condition. So that means that this is an experiment procedure. So we have the bacteria labeled with tempera dye. So this red dye attached to the trihalols is similar to the green one that doesn't have the beta lactamase moiety. So after labeling it with the dye, it will become red. And then we feed the bacteria into the macrophages to mark mark where the bacteria is. And then we put our own dye, the newly synthesized dual targeting dye, into the macrophage. And then we can do imaging to see if our dual targeting dye will improve the signal to background ratio, and also if they can specifically labeling the cell wall of the bacteria inside the macrophages. So as you can see here, that in the first channel is the green channel of the dye, and then the second channel is where the first dye is, the red tempera trihalols. So that marks the bacteria before the phagocytosis happens. And then the third channel is a overlay of the first two, and then we also did a membrane stain to see where the macrophage is. So in this case, that you can clearly see that only with 2 macromolar of dye concentration, we can clearly see the bacteria. And then, but if you use the same condition with the dye without the beta lactamase recognition moiety, the required concentration that you need is at least 100 macromolar. It's much more than that of what we much more concentration is needed. So that means that by using the 2 targeting strategy, our dye was able to achieve the phagocytosis bacteria labeling with a much lower concentration. So finally, we want to understand if the bacteria is being labeled on the cell wall, but not anywhere else. So in this case, that by labeling we zoom in of the bacteria in the macrophages with super resolution structured illuminated microscope to see the details of the labeling behavior. In this case, that we can see that the dye is clearly labeled on the cell wall structure of the bacteria, and this dye is specifically labeled on the polar region of the bacteria. So this is a unique behavior of Mycobacterium because of the uneven labeling and the division of the bacteria. when they split into daughter cells, they can inherit different cell wall material from the parent. And this is considered to be a unique niche for the bacteria to survive in the macrophages because of the uneven division and the inheritance of this material. So they have they could have different fighting strategy to survive in the macrophages. And our probe was able to clearly showing that that behavior, and this has not been quite achieved in the previous studies. So if you you see in the right channel, so those are the bacteria that is feeding into the or labeling before the phagocytosis, and that has been is what has been achieved in the previous study by but our probe was able to label these bacteria after their phagocytosis into the macrophages, and that is very unique for our probe. So finally, we want to see if our probe could differentiate live bacteria from the dead ones in the macrophages. This can be achievable because our probe is a metabolically labeled probe. So if the bacteria is dead, it shouldn't uptake any of our probe. So as you can see here, indeed in the live bacteria, you can clearly see the fluorescent signal in these bacteria, but in the dead ones, you don't see any signal. But these bacteria can be non-specifically labeled by the antibodies to let you see that indeed there are bacteria, but they are not being able to labeled by our dye. So in conclusion, that our dye was able to specifically label the live bacteria, not the dead ones. So some highlights and the future direction. So in this study, we rationally designed a probe with dual target lock strategy by using this two target in the probe. We demonstrate that our strategy gives higher selectivity for the Mycobacterial tuberculosis over other bacteria species. Finally, we demonstrate that our probe was able to label single live phagocytosed Mycobacterial tuberculosis, and this has not been quite achieved in the previous studies. Some future direction that we thought we could achieve is using this probe to do drug screening with intracellular Mycobacterial tuberculosis. As I mentioned before, that to understand the drug behavior in the for MTB in the macrophages and in their innate environment is very important, and this is quite different from when you're doing drug screening in the test tube. Second, we want to achieve in vivo MTB labeling, specifically using zebrafish as the animal model to understand their in vivo behavior. So with that, I would like to thank my advisor, the Jiang Hongrao and Professor Bertozzi, and also the funding sources. And if you have any question, please feel free to ask.